Hello student, like the previous one, we are back again with another chapter, that is biotechnology, its principle and processes. So in this chapter, we will discuss about the principle, about the tools, and about the process of biotechnology in this chapter, okay? And in this class, we will discuss about the principle of biotechnology and about how this biotechnology will take place. Now come to the point. So we human beings have been using these living organism, enzyme, microbes to produce product and processes which are useful to we human beings. That means we can consider this biotech as a technology, okay, where organism, microbes, and even enzymes are used to produce product and processes useful to we human beings. ꯑꯗꯨꯅ ꯕꯥꯏꯑꯣꯇꯦꯛ ꯍꯥꯏꯔꯒꯅ ꯑꯩꯈꯣꯏꯅ ꯃꯥꯁꯦ ꯀꯔꯤꯅꯦꯅ ꯀꯟꯁꯤꯗꯔ ꯇꯧꯕ ꯌꯥꯒꯦ ꯍꯥꯏꯔꯒ ꯃꯥꯁꯦ ꯇꯦꯛꯅꯣꯂꯣꯖꯤ ꯑꯃꯅꯦꯕ ꯀꯔꯤ ꯇꯧꯕꯩ ꯇꯦꯛꯅꯣꯂꯣꯖꯤꯅꯣ ꯍꯥꯏꯔꯀꯥꯟꯗ ꯑꯩꯈꯣꯏꯅ ꯃꯥꯏꯀ꯭ꯔꯣब्ꯁ ꯑꯦꯟꯖꯥꯏꯝ ꯑꯣꯀꯦ ꯏꯚꯟ ꯀꯩꯗꯧꯕ ꯁꯦꯜꯁꯤꯡꯁꯦ ꯁꯤꯖꯤꯟꯅꯔꯒ ꯑꯩꯈꯣꯏꯅ ꯄ꯭ꯔꯗꯛ ꯈꯔ ꯄ꯭ꯔꯣꯗ꯭ꯌꯨꯁ ꯇꯧꯕꯗꯣ ꯀꯔꯝꯕ ꯄ꯭ꯔꯗꯛꯅꯣ ꯍꯥꯏꯔꯀꯥꯟꯗ ꯑꯩꯈꯣꯏ ꯍ꯭ꯌꯨꯃꯦꯟ ꯕꯤꯡꯒꯗ ꯀꯥꯟꯅꯒꯗꯕ ꯑꯗꯨ ꯑꯣꯏꯕ ꯇꯥꯔꯒꯗꯤ making of bread, making of wine, making of curd, which are all microbe mediated process. Microbe ಧರ್ಮ ꯉꯥꯛꯇꯅꯤꯀꯣ ꯑꯗꯨ ꯑꯣꯏꯇꯥꯔꯗꯤ ꯍꯥꯏꯕ ꯄ꯭ꯔꯣꯁꯦꯁꯁꯤꯡꯁꯦ we can consider this as an example of biotechnology. Okay, but when we come to modern science, this biotechnology mainly deals with the modification of genetic makeup of an organism or for the production of chemical, for the production of antibiotic, and for the production of vaccine. Aduna modern science la kanda di biotech se mainly kaita deal to ge kanda organism amagi genetic makeup to modify to thopiba natraga koina chemical gumba enzyme gumba antibiotic gumba ko moise produce over large scale aduina mainly deal to. Okay, aduga European Federation of Biotech na moina biotechnology kamain define torong have to yingsi ko. So this European Federation of Biotechnology, they define biotechnology like this. Biotechnology actually it is the integration of natural science and organism, cell part thereof, and molecular analogs for production and service. So this is the definition of biotechnology according to European Federation of Biotechnology. Now come to the principle of biotechnology. So there are two main techniques that give birth to the modern biotechnology. One is genetic engineering and other one is bioprocess engineering, or you can call it chemical engineering. Okay, so first come to genetic engineering. So in this technique, for example, the genetic makeup of an organism, it may be DNA or it may be RNA. Okay, this genetic makeup will be modified. Okay, so definitely there will be change in phenotype as well as genotype of the individual. And if the technique is used for the manufacture of biotechnologically products such as antibiotic, vaccine, enzyme, hormones, etc., then the technique can be known as bioprocess engineering. Aduna biotech se principle ani lai aham principle sina genetic engineering haire. Genetic engineering dari kadogani hakanda organism amagi genetic makeup to kana modify to dene aduna organism to da my genotype ta phenotype ta homola ane aduga hariba process se akoina atopa way da kamaina akoina antibiotic gumba enzyme gumba hormones gumba vaccine gumba produce over sinaba taragadi ngasagi principle se kai korena we call it bioprocess engineering kawe adu taragadi ngasai kadoba biotech principle ani leire. One is genetic engineering and other one is bioprocess engineering. Okay, so here we have a small chart to show how biotechnology will take place. Now think that this donor is resistant to a particular antibiotic while the host is not resistant to that antibiotic. And now we are going to make the host cell resistant to antibiotic through biotechnology. And for this, first of all, we have to isolate the DNA from the donor. And this isolated DNA will be cut by restriction enzyme into smaller fragments. And from these smaller DNA fragments, the targeted DNA or the desired gene will be selected out. And this selected DNA is going to be transferred into the host. For that, a vector or a carrier is required. And this vector will be represented by DNA. And this vector will be again cut by enzyme, and in this cut portion the desired DNA will be inserted. Okay, and this joining of this DNA fragment into the vector is also possible because of presence of another enzyme, that is DNA ligase, and it will result in the formation of a combination of DNA, and this combination of DNA is called recombinant DNA, and then this recombinant DNA will be introduced into the host. Now this host cell will now become resistant to antibiotic because it has the gene for being resistant to antibiotic. And in the host cell, this recombinant DNA will be able to divide to produce multiple copies with the help of another enzyme known as polymerase. So this is how biotechnology will take place. aduna biotech se understand tonaba aina piriba simple sat se simahan understand ton honabiu. So it's the killer any color donor hypomaga host hypomaga. So the point is that donors it is resistant to particular antibiotic. On other side, host it is not resistant to that particular antibiotic. Or host resistant and first of all DNA extract cut down into smaller fragments aduga apamba DNA khandokpaneba kamba DNA nakanda which is resistant to that particular antibiotic aduga asigi DNA se akoina host option deni option ge hari kai daka tai carrier daka tare vector daka tai and this carrier or vector should be represented by DNA aru harba vector se masu katore enzyme na aru cut to portions da apamba gene to option baneba aru kanan join to birani DNA ligase have enzyme mana aduga sida form to be yansanaba DNA se we call it recombinant DNA ka web or sigi recombinant DNA se koin host introduce obane host introduce okandang hygiene do yawgre for being resistant to antibiotics. So definitely host colony will become resistant to antibiotic aduga host ka doge kanda host ki polymerase have enzyme sinaraga recombinant DNA ma divide torneba kai thoni my multiple copies produce ore kai thwa gene cloning thwe. So this is how recombinant DNA technology will take place. So during genetic engineering, there should be transfer of gene to create recombinant DNA, and there should be gene cloning also. And this genetic engineering is also known as recombinant DNA technology because in this process there is formation of recombinant DNA. Okay. Aduna sida haining is a karino kanda recombinant DNA technology to matamda important event umu thokane. Sigi important event hum se the first one da it is gene transfer. That means there should be transfer of gene from one organism to another. Second one that it is recombinant DNA, formation of recombinant DNA. Recombinant DNA tala ko yanjinaba DNA from the ko. This is one of the most important event during biotechnology. And last one is gene cloning. Sign apam gene do large number da produce my copies man produce so this gene transfer, formation of recombinant DNA, gene cloning, these are the three important events which should take place during recombinant DNA technology or genetic engineering. Okay. So come to another important point. So during gene transfer, what happen? A piece of DNA will be introduced into the host, but this piece of DNA will not be able to multiply, will not be able to replicate. Actually, it is because the piece of DNA which is introduced into the host does not have its own origin of replication. So here question is, what is this origin of replication? So original replication is a particular sequence of DNA that is responsible for initiating replication. That means if a DNA is to divide, then that DNA should have its own origin of replication. So on other side, the piece of DNA which is introduced into the host does not have its own original replication. That's why that piece of DNA which is introduced into the host will not be able to divide. Okay. But if we can insert that piece of DNA into the genetic material of the host, then definitely the piece of DNA will multiply and will be inherited along with the host DNA. This is because the piece of DNA has become part of host DNA. Which DNA? Which has its own origin of replication. Okay, only then the alien piece of DNA can replicate, can multiply itself in the host organism. And the hiding is here. Karino harakanda. Akoina host the piece of DNA ma introduce torkanda adugi piece of DNA do ama divide to ngamoy havene. Kaigi ngamdrino harakanda di magi oiba on origin of replication yaudeba. Hariba original replication have say particular DNA sequence ni come sequence na replicate to do initiate to na baneba adu yaudrabina a piece of DNA do ma divide to ngamoy adubu hariba piece of DNA se host ki DNA come DNA magi on original replication yabudo adu ko insert to ngamba targadi masana hai piece of DNA do divide to ngamani natra aduga next generation natra mathang cell da inherit to yarani have. Okay. And before we go further, let's see one more important point, and it is about plasmid. So this plasmid, actually this is extra chromosomal DNA structure which is present in bacteria, and it does not function as genetic material. Okay, and this plasmid has the property of self-replication. Okay, and as this plasmid can replicate, we can now insert the desired DNA into the plasmid, and it can be used to deliver the desired DNA into the host. Okay, now plasmid से करीनो है तरह का दी bacteria जाऊबा extra chromosomal DNA structure अमने DNA ni ko extra ba yauba which does not function as genetic material aduga magi property se karina kanda cell replicate to ngame that means it has its own origin of replication adu itaragadi ngasai koina plasmid da apam gene do hapsalaga apam gene do host introduce over ya ane hamne right plasmid na karina 800 carrier vector in 800 okay this is the point okay that means plasmid are extra chromosomal structure which can function as vector to deliver the desired gene or the desired DNA into the host organism. Now let's see how recombinant DNA technology is performed by taking one example. Okay, so here a bacterium by the name Salmonella typhimurium, it is resistant to a particular antibiotic. On the other hand, another bacterium by the name E. coli is not resistant to that particular antibiotic. So through biotechnology, we are going to make this E. coli resistant to biotechnology. Okay, so let's see how this process takes place. So for this gene encoding for antibiotic resistance present in the plasmid of Salmonella typhimurium is cut out and the cut piece of DNA is inserted into the plasmid of E. coli. So like this, the first recombinant DNA was produced. And this first recombinant DNA was produced by two scientists. One is Stanley Cohen and another one is Herbert Boyer. Okay. And this cutting of DNA is also possible now, and it is due to the discovery of one particular enzyme, and that enzyme is known as restriction enzyme. And as this enzyme can cut the DNA, the enzyme is also known as molecular scissor. So here in exam question can be asked like this: what are molecular scissor and what are the role of molecular scissor? Like this question can be asked. So after this, the cut piece of DNA will be joined with the plasmid DNA, which will function as carrier or which will function as vector. And the linking or the joining of this piece of DNA with the plasmid vector is also possible because of the discovery of another enzyme, and that enzyme is DNA ligase. That means DNA ligase will help to join DNA fragments. And the new combination of DNA, okay, which is produced in vitro is known as recombinant DNA. And when this recombinant DNA is transferred into the E. coli, the E. coli will definitely become resistant to that particular antibiotic. And not only this, the recombinant DNA will also replicate with the help of host polymerase enzyme and can form multiple copies. So this is how recombinant DNA technology will take place. Na sida hainingise karino haikanda ihan hanba recombinant DNA do kanana puthongge haikanda Stanley Cohen ga Herbert Boyer ga Sigi Stanley Cohen ga Herbert Boyer ga na ihan hanba recombinant DNA puthong homa tamda moina kari sinai jinama sinare kama jinno which is resistant to antibiotic and present in the plasmid of Salmonella typhimurium ar haribe jinno lothoga E. coli gi plasmid ta link to bune ngama thangi points na kaina kanta ngasai gi link to gaita di DNA ni to be cut DNA ka tom do ka ta dra aduna DNA ka to busu possible oire have ne kai to be possible oino ha kanta DNA ka to ngamba enzyme ama lerambagi and that enzyme is restriction enzyme aro high enzyme se DNA ka to ngamine ke koino we call it molecular scissor ka to kanda ngasai gi apama DNA do okoina vector am da haptene sigi vector sina apama DNA do host introduce udini utadi vector dong link to busu possible oido karigino hakanda another enzyme ama lerambagi adugi enzyme ke koina we call it DNA ligase kore aduga hauji form to ba yanjanaba DNA hai tare ko mapana form to ba yanjanaba DNA do apama gene ne apama DNA do ne plus kana vector DNA ne adu yanjanaba DNA ke we call it recombinant DNA or hybrid recombinant DNA do koina host E. coli hapjan mane hapsan taradi masana apama gene to introduce ograbani E. coli will now become resistant to antibiotic sidanatana ngasagi E. coli yoy kanano polymerase have enzyme am yauri sigi enzyme sigi mating loina kadare ma replicate tawani aduga magi multiple copies se produce obanga manhanib so this is how recombinant DNA technology takes place. So from this simple process, you can find out the tools of recombinant DNA technology. Okay, so here the tools are donor restriction enzyme, vector DNA ligase, host, and polymerase enzyme. Okay, and this tool we'll discuss in next class. So this brings me to the end of this class, and today in this class we have discussed about the principles of biotechnology and about the processes of biotechnology, how biotechnology will take place, and hope you can understand each and every point. And thank you very much for your attention. Okay, thank you.